This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1205, recorded on the 28th of March, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it is 57 Fahrenheit, 14 C, and not very pluviose, but it is supposed to get that way this weekend.
Yeah, it's supposed to start being pluviose today, actually. Saturday, Sunday, Monday, Tuesday. It's 18 C and cloudy here in Chelsea, which is New York, not the UK. Chelsea's a neighborhood in London, right?
I think so.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 62 Fahrenheit, which my phone says is 17 Celsius. And the weather picture on my phone looks much more cloudy and foreboding. I mean, it's cloudy outside, but that's like scary looking. So I think it will be pluviose here as well soon-ish.
Yeah, we're going to be using this word forever. It's from one of today's papers. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. I missed the reference to fluviose in one of today's papers, so I am as confused as the listeners are.
You'll see it.
It's 60 degrees Fahrenheit, feels like 51, which means it's 15 and a half Celsius, feels like 10 and a half. And it's kind of gray with a slight tinge of blue.
And pluviosity is raininess.
Yeah. And we'll see today in one of our papers what that has to do with
the word,
anything.
And it is relevant. I'm not entirely sure that it was necessary to use that word, but it's a good word.
Yeah, I kind of liked it. It's, the authors were from Brazil, right? So it's interesting that maybe there's a similar word in Portuguese. And
yeah, most likely.
That's why they did it.
Yeah.
It's in the OED, it says it's obsolete and rare.
word.
Flowing much.
Well, we're going to bring it back.
Yeah, we're bringing pluviosity back. Yes. It's a perfectly cromulent word.
Pluviosity. If you like these discussions about science, please support us so we can continue to do them. We do need your support because we don't run ads anywhere. So we have no source of income except your donations. So please go to microbe.tv/contribute. ASV, Kathy Spindler.
So this is not much different from what we've heard in the last weeks because they don't have the special workshop programs up yet, and they don't have the concurrent workshops up yet. They're still working on those. But the meeting, of course, is sponsored by or hosted by McGill University, and there are several other universities in Montreal, so there will be people from there as well. It's going to be held at a really beautiful convention center, the Palais de Congrès de Montreal, July 14th to 17th, 2025. And so the deadlines that are coming up really fast, it's almost April, are ASV Cares and Kitty Corp. So Kitty Corp is if you want to have your kids there on site. ASV Cares is if you want to apply for some kind of grant to take your kid's grandmother along, or take them to grandma's house, or have a daycare person come into your house so that you can attend the meeting. You have to write a justification and a budget and all of that. And for both of those, Kitty Corp on-site childcare and ASV Cares, you have to be a member. The deadlines for those are both the same, April 9th. So there you have it.
All right, some news. No shortage of news in science this week.
Shortage of good news in science. But,
yeah, there is.
Yeah.
Sorry, these are mostly New York Times links, but first one by Teddy Rosenbluth. The headline is, Remedies supported by Kennedy leaves some measles patients more ill. After the health secretary promoted vitamin A as a cure, parents in West Texas be giving their children high doses, sometimes to prevent infection. And now doctors are seeing these children with signs of liver damage because you can't take too much vitamin A. Now, it's a fat-soluble vitamin, concentrates in certain organs. RFK Jr. said at the beginning of the outbreak that vitamin A was a miraculous cure. And so this is highly unfortunate that instead of starting a vaccination program, he just stepped back and said, get your vitamin A.
So I have a comment. It reminds me and other people I've talked to about of injecting bleach or using ivermectin.
Yes.
But also the fact that Daniel has said on the clinical updates that there is, a scrap of evidence for this in developing countries where kids are generally malnourished.
Yeah.
If you give them some supplement of vitamin A under a physician's care, it can help alleviate some of the difficulties with measles, but it does not prevent the disease.
No.
And it is extremely it would be very unusual for a kid in the United States to have any significant vitamin A deficiency, I think. I mean, we're awash in food here.
So there's an interesting quote in this article from Kevin Griffiths, who until last week was the communications director for CDC. And he resigned because he didn't like how RFK was handling the outbreak. And he writes, in my final weeks at CDC, I watched as career infectious disease experts were tasked with spending precious hours searching medical literature in vain for data to support Kennedy's preferred treatments. Can you imagine? Wasting your time. All right. So then this is an article, actually, a science article in Pediatric Infectious Diseases that Daniel mentioned last night, but I think you should know about it. Vitamin A in children hospitalized for measles in a high-income country. So this was a 2021 study in Italy where they looked at 108 children who were hospitalized for measles, and half of them got 2 doses. That's the treatment, 2 doses of oil-based vitamin A, and the rest didn't. Vitamin A, their conclusion, does not change the clinical course of measles infection or the rate of complications in children hospitalized in a high-income country. As Kathy says, it does so in under-resourced countries where the kids are undernourished. So either this is willful ignorance or something else. Now, my colleagues sent me this one. Well, I can't get... Yeah, I can get it. I got one free article. It seems that every time I go to a science article, it says you have one free article left. Okay. Science
and
article.
I'll repeat my reminder that if you purge the cookies from your browser and go back, you can then read another article.
Yeah, maybe that's what I'm doing inadvertently.
Yeah.
By John Cohen, saying the pandemic is over. NIH starts cutting COVID-19 research. So people working on COVID got letters saying, it's the end of the pandemic. We're terminating COVID-related grants because those were issued for a limited purpose to ameliorate the effects of the pandemic. Pandemic's over. We don't need these grants.
Because we only have how many tens of thousands of people a year dying of this now?
And we haven't learned anything.
No,
and the thing is,
the research was totally wasted.
This is such a BS excuse.
Yeah.
So many of these grants are for prevention of future pandemics, for making antivirals and so forth. Oh my gosh, they're just lying about the actual reason for terminating. For example, this one program, which Charlie Rice is part of, looking for antivirals for bunyaviruses, filoviruses, flavies, paramyxopicornos, togas. Charlie says the termination of the program has a misleading rationale and is a pointless, ill-advised move that will hurt U.S. science and pandemic readiness. So this is just, okay, the flu pandemic was over years ago. We're going to terminate all flu research? What are the pandemics?
They'll take that idea.
Yeah, well, I heard that flu is one of the few things that's going to continue. But this is ridiculous. But we shouldn't be surprised. Every day there's many ridiculousness. Like the Queen said to Alice, why every day
Oh,
believe 7 impossible things before breakfast.
Right.
Yeah. Once upon a time, someone gave me this gag gift of this little doll that you're supposed to hit on your desk when you're mad to help you feel better, and it now has to live permanently here. So that when I hear about all of these things, it's ready to go as I just sit and read news articles like this.
Another one in Times by Stephanie Nolan, the U.S. to end vaccine funds for poor countries. So they got a hold of a 281-page spreadsheet that lists their, this is part of USAID to terminate foreign aid programs. And so they're cutting off vaccination programs.
Health problems elsewhere will be our health problems very soon.
Oh, yeah.
There's no border you can make that keeps out microbes.
Nope.
Well, they don't seem to get this. And, a related thing, I'm not going to link it. There's too many articles, but there was another article in the Times just yesterday. Musk has claimed, the title was Musk claims nobody has died from his USAID cuts. And that's a lie. And they go through names of kids who, for example, their HIV was in check because they got it at birth from their mother. This one kid was, 8 years old. He'd been living all these years. The U.S. supplied him antivirals. No more antivirals. He died in February of pneumonia. And all other kids, lots of other stories like that.
And that's just in the month that this has been happening. I mean, this is not something that's a one-time event. This is going to be ongoing casualties worldwide till we fix our government. Yes.
I was just looking around. All I could see was my antibody.
It really helps.
Just cannot handle the lies. That's the thing. But also, why be so mean? Who else? I mean, other people may step in, other countries, and hope they do to help these people. But why can't you help? This program, like PEPFAR, was started by George Bush. He was a Republican. Oh, my gosh. Yeah, okay. And then one more. I mean, it just gets worse. Look at this. Is this a... No, this is not. This is a science-based medicine article by Stephen Novella. So RFK Jr. hires David Geyer to do a study on vaccines and autism. There have already been over a dozen. There's no connection. And so he hires this guy who is not a doctor. He's not a scientist. He's an anti-vaxxer. So obviously they're going out to try and prove that vaccines cause autism, which is not the way you're supposed to do it, right? Oh my God, this is a really good article. You should read. And he said, we're not going to, this guy Geyer has a history of publishing horribly, fatally flawed studies of vaccines. They are the kings of the dumpster divers. And they have promoted dubious treatments. So he says there's nothing in this study that will be useful. Here's what the study is going to be. I'm going to tell you. They will find 1,000 kids with autism. They'll find that they're all vaccinated. And he'll conclude that vaccines cause their autism, which, if you know anything about science
or logic,
is a flawed conclusion.
Yeah.
Right? But he will publish it. RFK will trumpet it. And, people will believe it because it comes from a supposedly...
Can I be the reviewer?
I mean, it's not going to be published in Science, Cell, or Nature, that's for sure. It's going to be in some obscure place because they will never take it. Okay, Kathy, you had an interesting addition.
Yeah, a listener sent this in. She's in another department here, so thank you, Sue, for sending it. I don't know if this was already in the works or not, but there's a new policy that's going to happen on May 6, 2025, new federal policy for oversight of dual-use research of concern, DURC, and certain other research with pathogens that will go into effect and will apply to all U.S. federally funded research. And so the U.S. government policy link is here, and then there's a link from the U.S. government about implementation guidance and an NIH funding agency implementation guidance link. So bringing this to the attention of the TWiV listenership, that might be in the United States and might have federal funding, our institution has been proactive and is getting from everyone by a short 5-minute or less questionnaire about what pathogens and biological toxins you work on. So doing a risk assessment so that they can have that on hand and be ready with whatever comes down the pike May 6th or 7th or whatever. And so you may want to look at these federal links and figure out if there's something that your environmental health and safety people or whoever you have are on, and get it going.
Soon we won't even be able to talk about these kinds of experiments.
Yeah.
I got an email from someone who said, "You're encouraging people to do gain of function. You should be thrown in jail." Seriously?
Wow.
That's not how this works.
No.
Yeah. Wow.
This is a very, very suspicious situation here. You try and muzzle the people who know better, right? I wonder what that is. All right. Now for some science.
Yeah.
Much more fun.
Our snippet today is in Nature Medicine. You want to summarize that for us, Kathy?
Sure. The title is Jamaican Fruit Bats' Competence for Ebola but Not Marburg Virus is Driven by Intrinsic Differences. And I didn't check if there's any co-first authors. Doesn't look like it. First author is Sarah Van Tol, and the final author is Vincent Munster. And so this takes place at NIH at Hamilton, Montana, also Braunschweig, Germany, and where else? Cornell, and more Hamilton, and NIH, and Bethesda, and Fort Collins. Okay.
Yeah.
So, so the filoviruses, there are 5 genera, which is the plural for
genus.
I had written here geniuses. Typo, and I was like, this is not making sense. Okay. So of those 5 genera, you're probably familiar with the 2 that we just heard in the title, Marburg and Ebola. And the focus on this paper is on 2 of those. And among all of these filoviruses, their reservoir species in bats, or their specific kind of bat, has been identified. But that's not true for Ebola virus. And so that's part of what they're going to be looking at in this paper. So there are 1,400 or more than 1,400 species of bats. And so the fact that one hasn't been identified as a potential reservoir host for Ebola could be that they just haven't tested the right one yet. Or it is also possible that bat is maybe not the reservoir host for Ebola, for most Ebola. So in this paper, the authors use Jamaican fruit bats, and that bat species is identified as a potential host for Ebola, albeit the fact that the bats get a non-lethal disease with mild clinical signs, but they do shed infectious virus. And that's in contrast to Marburg. If you infect these Jamaican fruit bats with Marburg, it doesn't do much at all and doesn't shed infectious virus. So they use a variety of approaches with these technically challenging viruses, which, if you're going to use the wild-type virus, are BSL-4, to study Jamaican fruit bats in the laboratory. So this is a tour de force. They identify some host differences in vitro that correlate with infection results when they infect bats. And so they find that the Marburg virus is less able to counteract the host type 1 interferon response. And so because it can't thwart that host response, it replicates less well, whereas Ebola has a way to deal with that, and so it replicates better. So anyway, they call this match of host bat species and virus complementarity, which kind of threw me because I couldn't figure out where that was coming from. But eventually they used it enough times that I got it from the context. And so doing this allows them to figure out ways to identify potential other bat hosts for other Ebola viruses or other filoviruses. It also provides a nice, useful infection model for Ebola virus and it has the potential to allow them to answer a lot of open questions, some of which they enumerate at the end of the paper. And one thing that I found really nice about this paper, and it kind of drove the way I read the paper, is that every figure legend title is the same as a subject heading in the text for each paragraph. And so it's really easy to match up the figures with the section of text that you're reading. I really like that.
What I liked is they do a lot of plaque assays.
Yes.
Yes.
They actually measure infectivity.
Yes.
Yeah.
And in many cases correlated with PCR measurements of RNA. So for a BSL-4 pathogen, it's not so trivial to be doing plaque assays, right?
Yeah.
And a BSL-4 pathogen where they are actively doing infections of bats. They have a bat colony, which very few people do. So yes, it is possible that Jamaican fruit bats are not the actual host species of Ebola. Perhaps it is a bat that's found in Africa. But there are almost no situations where people have laboratory bat colonies. And so setting up this model system, as Kathy mentioned, where you can actually study Ebola in live bats, and then looking at how those results correlate with or relate to what you see in a cell culture model is super important.
And I just want to point out there is a probable reservoir host or believable reservoir host for Marburg virus in the Egyptian Roussette bat, which they also talk a bit about. So Marburg can grow and replicate and be released from these Egyptian Roussette bats, but not the Jamaican fruit bats. The Jamaican fruit bats can support Ebola virus infection, but not Marburg infection. And so it provides, a nice comparison set that they can work with now that they've got their bat colony and their BSL-4 and all these techniques, and look at really what they're trying to get at is what are the factors that determine what reservoir host these viruses can grow in, because that's really what we'd like to know.
I looked it up, and Jamaican fruit bats aren't in Africa. So it's a model system.
Yeah.
But the Egyptian Roussette bats, well, obviously, if they came from Egypt. But there were, I don't know if it's the same bat, but there were examples where they were able to trap bats and find infectious Marburg virus in them. And I don't know if it's the Egyptian Roussette or another species.
I think it's the Egyptian Roussette.
Roussette, yeah. Okay. Just want to point out 4 of the authors have been on TWiV. Tony Shounts, Raina Plowright, Andrea Marzi. No, Andrea was not on, sorry. And Vincent Munster. Yeah.
Yeah, and it's a single first author, Sarah Van Tol, and a single senior author, Vincent Munster.
Some of these other viruses are interesting. Raven virus, RAVN.
Right. So the paper is
Marburg.
open access, and the intro to it is actually a really good survey of filoviruses and bats.
Yeah, I really liked that part.
So Raven is related to Marburg. And
Mengla.
Mengla.
Which
I like that one
we
too.
did one time because I can still hear. I played it over and over on the Google pronunciation thing. Mengla. Mengla.
Well, and we also did one on another virus I talk about, which is LLOVIU.
Love you.
Love you.
Which we called Love You. A friend of mine later was like, that would not be how you would pronounce that in Spain.
Well, the guy who was on, who found that in Schreiber's bats, I forgot his name. Well, he was from Hungary anyway. He called it Love You. Yeah. And so did the lady at in Boston, the BSL-4 lady. Yeah. She called it Love You too. Yeah. Oh, that's fun, right?
Yeah, absolutely. So
it's easy to remember.
However one wants to pronounce that word, we've talked about it on TWiV.
And I think in Wales it might be Love You or something like that.
Yeah. So that was isolated from European bats, and then it was found in Schreiber's bats, but doesn't seem to be any connection with infection and death. There's also one in China recently found De Hong virus. This is the newest member. It's from Leschenault's Rousset bats in China. It seems to be circulating within viruses in that country. Mengla. It's cool with some interesting accents on two of the letters. Bombali virus. That's from Angolan and little free-tailed bats in Sierra Leone. Extensive distribution within Africa. But they say, aside from Bombali, there is little evidence for these Ebola viruses, ortho-Ebola viruses circulating in bats. There's some serological data. This is interesting. Serological data support that several bat species, including the hammer-headed fruit bat, are exposed to filoviruses. Oppositionally, I would have said in contrast, but that's fine. A study that evaluated viral signatures of reservoir hosts predicted that Bundibugyo and Thai forest, two Ebola viruses, circulate in a primate host. Wow. And they say maybe we're looking in the wrong species. We're looking in bats all the time for Ebola viruses, but maybe that's not right.
Yeah, so one issue could be that we're looking in bats, and that's not the right species. The other issue could be that perhaps these bats are only infected with an acute infection for a short period of time. So we're catching some who were infected with that serological evidence that we haven't happened to pick the right species on the right day that bat happens to be infected.
Right.
I don't know, maybe it's sick and staying home that day.
Yeah. Yeah. All right, anyway, they have a colony of Jamaican fruit bats.
Oh, before you go to that, because the paragraph about the 1,400 species of bats mentions the two bat suborders. I don't know if you picked up on this, but there's the Yingteroptera and the Yang. So there's the yin and yang. Yeah.
Yes.
I thought that was
cool. I didn't notice that. Filoviruses can infect bats from both suborders. And these diverged 63 million years ago. So maybe filoviruses were infecting the common ancestor. So they have a colony in Hamilton, Montana of, is it in Hamilton or is it in
I think
Fort
this
Collins?
is the Fort Collins colony.
But there's no BSL-4 in Fort Collins, is there? Oh, yeah, there is, but
I'm not sure.
I get the feeling that this was done in Hamilton, Montana.
When I looked at the methods, it looks like the bats were at Fort Collins.
So they infected them there?
Or transported them to Hamilton.
Yeah, either way. So they have a colony of... Yeah, I went and looked at this colony with Tony.
So yeah, Jamaican fruit bats from the colony housed at Colorado State were used upon arrival at RML. So they must have been grown at Colorado State and then transferred to Hamilton.
So they're about this big. I saw them hanging on the ceiling, right? 6 inches long or so. And Tony and I walked in the room and he said, "Watch out, they'll pee on you." So what am I supposed to do?
Wear a
But
hat.
they didn't. They were just flying around, and the tables were covered with slices of melon for them to eat. It was very cool. They were hanging in clusters from the ceiling. So they take these bats, and they take 12 for each virus, and they inoculate them with Ebola or a Marburg virus, oral, intranasal, and subcutaneous. So all three routes at once, because they don't know which is going to work, and I don't think they have the number of bats to do
Bats are probably not cheap to work
all
with.
three, right? So you need 36 per virus if you wanted to do all the routes. So they say later, we need to go back and do it individually. But
Find the best route, yeah.
Then at 7 days post-infection, 4 of each bat were euthanized, and then the rest were monitored through 28 days. The Ebola-infected bats developed mild hypothermia. Their temperature went down. And then they returned to baseline by 28 days. No changes in body weight. The Marburg-infected bats didn't have any change in body temperature or weight.
So the hypothermia was kind of interesting because, humans with Ebola would spike a fever, hyperthermia. And I was wondering if this is typical of sick bats? Because they normally have a high body temperature, do they lower it to try and combat infection, or is this?
I am not sure. I will go back
don't
through
know.
all of my bat info that I have.
Yeah.
I don't know. So lymphopenia is a hallmark of filovirus disease in some hosts. So they looked for that, but they didn't see any changes in white blood cells in either Ebola or Marburg infected bats. In Ebola, the number of monocytes increased significantly, but that's the only difference that they saw. Then, of course, they collected oropharyngeal and rectal swabs to monitor virus shedding. And they could find Ebola. They did it by two ways, PCR and infectious assays. Actually, they did TCID 50, so it's not a plaque assay, but it's an infectivity
They
assay.
looked at
so
infectious
it's good.
virus.
It's good. Marburg RNA. So Ebola RNA was detected in oral swabs and rectal swabs. Marburg RNA was not detected in any swab. So they're not shedding any virus. And no infectious Ebola was found in oropharyngeal swabs, but not in rectal swabs. And no infectious Marburg was found in the oropharyngeal swabs. Okay, you're getting a picture for how these two differ so far, right? Looks like Marburg is a little bit anemic in these.
Looks like this is a dead end host just dead end for Marburg. It doesn't really get going at all.
Then they looked in various tissues to see where the virus is spreading, and they could find Ebola RNA everywhere. Well, they looked at the skin, at the inoculation site, salivary gland, lung, liver, spleen, kidney, reproductive organs. They found Ebola RNA everywhere. Highest were in the skin where they inoculated it. No surprise there, right? And they could still find Ebola RNA in spleens at 28 days. And they say the titration of the tissue samples largely reflected the RT-PCR data with infectious virus in at least one sample for all tissues tested. Minimal Marburg virus RNA at the skin where they inoculated, and didn't get anything. Very, very low copies in liver, spleen. Everything else was negative for Marburg. And minimal infectious Marburg at the site, at the injection site, and livers. Okay. Yeah, they look for signs of inflammation by histopathology. They see some inflammation in Ebola-infected bats in the skin. Sometimes neutrophilic lymphadenitis in cervical lymph nodes. They see some changes in the spleen. But in the Marburg-infected bats, they do see a little bit of histopathology changes, but nothing significant in the organs. So even for both, I'd say it's probably minimal histopathological changes. All right, so then they turn to the responses of the host, the innate responses and the humoral responses. And they just the hosts respond. Let's put it that way.
Yes, the hosts
because they
respond.
just make a list of all the things that go up, and I don't find it extremely useful. I would have liked a summary, but we don't have one. But they look at gene expression, right, on the different tissue samples, looking for interference-stimulated genes and inflammatory cytokines, and they're induced significantly in Ebola-infected bats, and they remain elevated at 7 days. That's both cytokines, inflammatory cytokines, and interferon-stimulated genes.
Yeah. And when I look at these data, they're looking at basically day 3 and day 7, as we've mentioned, following infection. And overall, I see that there seems to be more going on with cytokines when the Ebola infection. There's higher amounts of cytokine than there is with the Marburg infection.
Yeah. Yep. And for Marburg, in fact, in lung, there's nothing at all,
Right.
whereas there was in Ebola. And they also do transcriptional analysis, looking at genes up and down regulated. So Ebola infected bats, 241 upregulated and 460 downregulated genes, mostly involved in regulation of the immune response, and less so in Marburg infected bats. Okay, so you get innate responses to both, but it's much greater for Ebola virus.
Mm-hmm.
Then they look at antibodies, and they're using ELISAs, so they're looking for binding antibodies. And all four of the Ebola infected bats make Ebola G protein specific antibodies, and they're pretty nice titers here. And only one of the four Marburg infected bats seroconverted. So I guess that's in line with the low reproduction of that virus in the bats, right?
Yeah, I mean, this is one of those things that I actually had to spend a little time thinking about some of the stuff in this paper because I read it not as deeply as I could have the first time and confused myself greatly, and then had to go back and spend some time. And so I think that this part of the paper overall is sort of summarized with this idea that when they look in the in vivo situation at days 3 and 7, they see more of an immune response in the Ebola infected bats than in the Marburg infected bats.
Mm-hmm.
And so here it's sort of like Ebola is inducing a larger immune response. And then that will sort of come back into play when we see some of their later data.
They also looked at spleens and lymphocyte populations. The total T and B lymphocytes in Ebola infected bats didn't differ from healthy controls. And I'm guessing it didn't differ in Marburg infected bats either, because they barely mount an infection. They look at the frequency of VH and J gene segments, but it seems to me that, well, you get increases in Ebola, but not Marburg.
Yeah. Basically, they're seeing this big immune response in Ebola, or relatively big immune response in Ebola, and not so much in the Marburg infected bats at days 3 and 7.
Okay. Then they previously published some work. They have cell lines or cells from Jamaican fruit bat, and they had published a paper saying that reproduction of Marburg seems delayed compared to Ebola. So they want to look at this further. So they look at 3 Ebola strains and 3 Marburg strains in viro cells, vervet monkey kidney cells. Is that right, Brianne? Vervet or Grivet?
I believe it's vervet.
You know, I ask you this all the time, and neither of us remember.
I know.
Right?
I'm double checking right now.
And then they have primary Jamaican fruit bat cells. I wonder where the Uropathogeum derived. What is a Uropathogeum? Does anyone know? Oh my gosh. Let's look it up.
Sounds like an infectious disease conference somewhere in Germany.
The Uropathogeum is the membrane of the skin that extends behind the hind legs and tail of some bats. And now, I see. So they cut that up and make cells out of it.
Oh.
Interesting. We learned 2 words today. But I'm never going to use Uropathogeum again.
No, that doesn't come up much.
Doesn't come up in daily life.
It comes up sometimes on trivia questions.
Would that be a Jeopardy question?
I have not seen it on Jeopardy, but I have seen it in other online trivia.
Ah.
God, it would be a stumper, right?
Mm-hmm.
Titers of all the Ebola strains on the JFB cells were within 20-fold of the titers on Viro. The Marburg were 2,000 to 32,000-fold lower on the bat cells than on Viro. So they have a problem with reproducing in bat cells, I would say.
Yeah.
Is that a fair conclusion?
So Marburg is not reproducing particularly well in these cells.
No.
And yes, it's vervet, by the way.
Vervet? Okay.
Yeah.
Thank you. So I will remember that because V is the first initial of my name.
Yes.
We'll do that.
And Vero and Vervet.
Yeah, that's even better. All right, so why is this difference? This is interesting. This is a fundamental virological question, right? So first they said, well, does it depend on virus entry? So they make some pseudotyped viruses with vesicular stomatitis virus that do not replicate. So in other words, you put the glycoprotein of the Ebola or the Marburg onto the VSV, and then it will enter cells via the glycoprotein. But this you can do at BSL-2 because there's
Makes things a lot more convenient.
makes it easy. And you use GFP, I think, as a to measure reproduction. I think I may be wrong,
You
but
usually
anyway,
do,
you can measure it. Yeah. So the Ebola pseudotypes entered more efficiently than the VSV pseudotypes, but when they use infectious pseudotypes, they all grow to comparable titers. But then they say when they normalize the infection ratio, it turns out that VSV Ebola grew to a 13-fold higher titer than VSV Marburg on these Jamaican fruit bat cells. So this is
Marburg
VSV.
has some kind of an entry problem here.
So this is focusing the difference on the receptor interaction because the VSV is the same for both, right? So you could say, yeah, the early events make some difference, but then they leave that; they don't come back to it anymore, and then they turn to interferon signaling. So they infect these Jamaican fruit bat, and they also have Egyptian rosette bat cells. They can do these experiments. So you can't really do these in bats, right? Look at interferon responses and so forth, and specifically dissect them. So they infect these two cell lines with Ebola or Marburg, and they look at activation of the innate antiviral response. And they find, again, that Marburg lags behind. Ebola replicates to a higher titer, but still Marburg induces phosphorylation of STAT1 and 2, which is an early part of the induction of the innate response and also expression of ISGs. But so at 72 hours, whereas Ebola infection did not activate through 120 hours.
Yeah. And so I think that, so this is the spot where I confused myself and had to go back for a second. Because here they're looking at interferon stimulated gene transcription, and they were also looking at that in that previous figure.
Yeah.
But here they're looking in cell culture and they're using some earlier time points, whereas in the other one, they're looking at full bats at days 3 and 7.
Yeah.
And so I look at these data and I see that Marburg seems to be inducing a very early interferon response, which may help to stop Marburg replication. So that may, in fact, further reduce replication in addition to some of these blocks that we're talking about in terms of entry. And exactly where the interferon and where the entry fall in, we don't know. That we could argue about. But the idea is that Marburg is tripping the immune sensors early, and thus that immune response is diminishing the amount of virus. And so then later on, there's actually less of an immune response to Marburg because there's not as much virus around.
Yeah.
So later on, there's going to be more immune response to Ebola because there's more Ebola virus to drive the immune response. But very early, Marburg is making more of a response, which blocks Marburg replication. So then we don't sort of get that response more amplified. Does that make sense?
Yeah. So there's a small part of the difference between the two viruses that is attributed to entry, but there's a big part attributable to innate responses.
Yeah.
Yeah.
So
it seems like Marburg is struggling to get in the door and then sets off the alarm when it does.
Yeah.
Yep.
So it would be interesting to look. Obviously, these are primary bat cells, so we have no knockouts. But if this were a mouse, they could look at interferon knockouts, but if they could do it here, I bet that would, if they could knock out the interferon pathway, that would probably restore Marburg replication in these Jamaican fruit bat cells. I would bet.
Potentially, yeah.
But it's primary cell, so you can't really. I don't know. There are probably ways, and I'm sure they're working on it because it would be interesting. So they come up with the idea that Ebola is making some. So the filoviruses are known to make innate antagonists. Okay, and they both do. Ebola and Marburg genomes encode innate antagonists. So the Marburg the protein is called VP40, and Ebola is VP24. They both block activation of the interferon response, but at different points. So they're thinking maybe that's the difference. Maybe one doesn't work as well. Maybe the Marburg one doesn't work.
Doesn't work specifically in the Jamaican fruit bat cells.
Right.
Yeah. And
It
essentially,
must work somewhere.
Yeah, it wouldn't be there if it didn't work. That's right. But I suspect that these are not the natural hosts, so
Right.
it doesn't really matter. And so they basically find that if you use an inhibitor of one of the kinases involved in interferon signaling, it's called itacitinib. Marburg titers increase, but have no effect on Ebola. So it's kind of like doing a knockout, right?
Yeah.
You do a chemical inhibition. And so that's suggesting that, in fact, Marburg can't antagonize, so it's eliminated. So it's a very interesting study. And it shows you that host differences are important for these filoviruses, and in particular, innate immune responses. And you can use this, and now they have two viruses that do very different things in the same bat cell, so you could tease it out further if you wanted to. Right? You can understand precisely what's going on there. Let's see, what else do we learn here? Oh, so they say we can use this to interrogate a plethora of open questions. Okay.
This is just a good vocabulary, TWiV.
I would just say answer a number of unanswered questions, but that's because I don't write well, I guess. So the contribution of the host immune response, which you saw here, obviously innate responses, but they could dig deeper in that. And another one is interesting, whether virus persists and recrudesces. There's another one, recrudesces when a chronically infected bat encounters different stressors. Oh, just go to Washington, bats, and you'll get stressed out. So, in the wild, bats are under all kinds of stress, like pregnancy is one, extended flight is another one. And this can influence virus reproduction levels and how ill they get and how much they shed and when and so forth. So they could use this. They do like the word interrogate. Now they say they're a valuable model for interrogating filovirus biology. When I was a kid, I'd never heard of interrogate, except in the context of
Police dramas.
police
Yeah.
dramas. Yeah. Now it's the rage, and the
Everybody's
English
interrogating
language
their data now.
should be a flexible language, but I also am allowed to complain about it. Interrogate. You won't hear me use interrogate. You guys like this paper?
Yeah.
Yes.
Mm-hmm.
Yeah,
Okay.
cool paper.
Good. All right. The next one, Kathy. I picked this because I know you like this virus. How do you pronounce it?
Well, I found for the first time a paper that describes how it's pronounced. I've always pronounced it Oropouche, and that's the way this paper that I found pronounces it. But there's a whole cadre of scientists who say Oropouche. So it comes from a place in Trinidad where the first virus was isolated in 1955. So there's a river, a wetland, and a third thing that have a village that have that name. So where's my copy of the paper so I can give you the title?
So I can actually do title and authors, and because I want to comment on something here. I think the folks at Nature Medicine must be listening to TWiV's complaints about having the see the end of the paper for the affiliations. So now they've just given up, and it's the list of the authors and their affiliations appears at the end of the paper. So they move the authors down to the end of the paper.
Well,
too.
Nice.
But
Nice.
I printed the paper because I'm a dinosaur. I always do. And then I printed the page that has just their names. And that would take up the entire
Yes.
first page,
Yes.
and so then if you included their affiliations, then you wouldn't even have the abstract on the first page.
So this is Nature Medicine. It's closed access, unfortunately, but the title is Human Outbreaks of a Novel Reassortant Oropouche Virus, or Oropouche if you prefer, in the Brazilian Amazon region. And the authors and their affiliations. There is a single first author, Felipe Gomes Naveca, and a single senior author, Gonzalo Bello, and a whole bunch of authors, as Kathy just explained, in between those two, and they are from a whole bunch of places, which, let's go ahead and get into this. Instituto Leonidas e Maria Deane Fiocruz, Manaus, Brazil. Instituto Oswaldo Cruz in Fiocruz, Rio de Janeiro, Brazil. Fundação Centro, I'm butchering the Portuguese, I'm sure.
I think you could just say many, many places in Brazil.
Many
Manaus,
places in Brazil.
Recife, Fiocruz.
Yes. Boa Vista.
And then you get to the
Rio.
end. Yeah, Rio de Janeiro.
Rio, Porto Velho, Rio Branco, Curitiba, Brasilia, or Manaus,
and then they get to the ones at the end that you can pronounce.
Ananindeua, and then we get to GISAID Global Data Science Initiative in Munich, and then some more Brazilians, and then the Reference Center for Tropical Infectious Diseases in Hamburg, and then we have UC Berkeley and Cornell University.
So, Kathy, what is the etymology of the word? Do you know? I mean, Trinidad. I
Oh,
don't know. Do they speak French there? Or?
No. This paper by Fred Murphy says it's Amerindian, and the ancient meaning of the word is not clear.
I see.
So,
because in French, pouche. Oh no, that's another word. Poche is pocket.
yeah.
So, and I thought oro was mouth, but I don't know.
Yeah.
We'll just call it orov.
Right. Orov. Yeah. So, and we have discussed this virus before, briefly. It was that there was a news story in February a year ago about the outbreak that relates to today's paper. That was TWiV 1087. And then, so that was February. And then in August, Daniel Griffin, in his clinical update, talked about sloth fever, which was about 21 U.S. residents returning from travel to Cuba who came down with sloth fever, which is Oropouche virus. So this is a virus that's found primarily in Central and South America, especially in the Amazon region. It's transmitted by midges, so that makes it an arbovirus. And it causes fever, nausea, vomiting, aches, other flu-like symptoms, but rarely death. Daniel commented on vertical transmission in pregnancy that can cause fetal death and congenital malformations. And the illness lasts about 2 to 7 days, but it can recur. And there's no treatment and no vaccine. So the molecular biology, it's in the family of the Peribunyaviridae. It's in the genus Orthobunyavirus. And it has segments, L, M, and S, which are very nicely standing for large, medium, and small. And it's negative stranded. And so it might start to sound to you like an influenza virus. And like flu, it does cap snatching to add the caps to its 5' ends of its messenger RNAs. But unlike flu, it doesn't do it in the nucleus because the bunyaviruses do their whole replication cycle in the cytoplasm, like any good RNA virus would do. So the title has the word reassortant in it. So the fact that there are these 3 different segments is a way that they can generate new viruses in the same way that flu viruses can reassort. And so that contributes to some of the evolution of the viruses, in addition to them being RNA viruses and having high mutation rates. So this outbreak in the Amazon region, so a year ago, I think the number was somewhere around 1,000 cases, and now there's more than 6,300 cases. And they sequenced almost 400 isolates from around this whole Amazon region, which is large And I wrote down, 2 million square kilometers. And so they sequenced about 400 isolates, and that's why they have all these authors, because they probably helped generate and provide the samples. And they found that the virus is a reassortant. It has one segment from one lineage and its other two segments from another lineage. And they go through and do a whole lot of phylogeographic reconstructions, and they have a proposed reassortment scenario, which isn't just one from this virus and one from that virus, but there's several other intermediate viruses. And so that's a proposed way that came about. And they have this geographic data that allows them to sort of propose how the virus spread from place to place in Brazil.
Yeah,
So,
because they know that mosquitoes can only fly so far.
yes.
And so
And midges.
And midges. Oh, I'm sorry, midges can only fly so far.
So I wanted to point out this virus is not that picky about its vectors. It can be spread by mosquitoes or midges, and may do differently in urban versus forest environments.
Well, whoever can only fly so far.
Can only fly so far.
And so if they look at the geographic distribution of some of this, as Kathy points out, they could determine, well, was this a movement of one of these arthropods, or was this perhaps movement of people?
Right. So if it's a short movement, it was probably a bug, and if it was a longer movement, it was probably people. And they also correlate this with seasonality. The case counts go up in the wet season, come down in the dry season, so it correlates with pluviosity. And the particularly notable thing about this epidemic, so there are occasional spillovers and epidemics of Oropouche virus. This one has been unusual in that they last for one season and then they end, and then you don't see it again for some time. This one has gone on for 2 seasons now, 2 wet seasons, and it is also covering a much, much larger area than past epidemics.
And I first became aware of Oropouche virus in 2008 when I met Eurico Arruda, who works on this virus, and was telling me then, this could be a very bad thing someday. And then in the meantime, we got chikungunya and I guess Zika that kind of displaced it in terms of visibility or whatever. But now Oropouche is coming around. So it sounds like it's pretty nasty to get it.
So we have midges here in the U.S., right?
Sure.
Mm-hmm.
So it could potentially come here. I thought people should be aware of this.
Right. And in fact, it used to be BSL-3, but I think in part because I rallied about this for a long time, but also because of the midges and midges already being here. It's now, well, as we're going to maybe get to the letters, it's risk group 2. So it can be, research now can be done if your institution evaluates the risk and determines that it's fine. You could do it at 2.
And by the way, there's a biting midge found on the Carolina coasts that is named after my family, Culicoides dovey. I don't know if that's still the current classification, but that was
Cool.
So this virus is maintaining a transmission cycle among vertebrate hosts, like sloths, as Kathy said. Daniel called it sloth fever, rodents, non-human primates, birds, by different vectors, variety of mosquitoes. And human infections are not needed to maintain this virus in nature. But occasionally it does spill over into people, then you get outbreaks. And when it does, the transmission seems to be driven mainly by midges.
So it has a sylvatic and an urban cycle.
Yeah. And also some mosquitoes. There have been about at least 30 outbreaks in the Amazon since the 60s that have been reported anyway, right? So the largest outbreak was reported in the Pará state in the 70s, 100,000 human cases. And then more recently, smaller outbreaks in Manaus, which is the capital city of the Amazonas state, in a rural community and in other rural settings. But they say that the burden of OROV infections has probably been underestimated because they don't have good laboratory diagnosis.
And because this
and-
is not generally lethal, people
Yeah.
get sick somewhere in the Amazon region, and then they get over it, and nobody hears about it.
And then, as Kathy mentions, there seems to be a recent expansion of the boundaries within Brazil. Between 2022 and 4, there's a sharp increase in cases in the western Amazon region, in both rural areas and major urban centers. And it was the largest upsurge in the 21st century and the most geographically spread epidemic, which is the subject of this paper.
And they refer to it as the Amacro region, which is the 3 Amazonian states of Acre, Amazonas, and Rondônia.
Okay, so but they look at the temporal pattern of the cases in Amazonas, where they did a lot of testing between November 22 and March 24. Small wave of cases between late 22 and 23, and then a major wave starting in October 23, which dominated until March 2024. And these peaks, as has been said, coincide with the rainy season. The virus infects individuals of all age groups. And as you know, the symptoms, Kathy has already told you about those. No fatal cases. So they have done a lot of sequencing to understand how this epidemic has been driven. So they sequenced full-length genomes, OROV genomes, from 382 patients in 37 municipalities of the Brazilian states of Amazonas, Rondônia, and Roraima between August 22 and February 24. And then they added those to all the known full-length OROV genome sequences that are available in databases. And they did phylogenetic analyses of those, and they find 2 large viral lineages in this outbreak sampling, which they just did, right? 2 viral lineages for the M and L segments, but the S is fuzzy, not clear where it came from. They say sequences were dispersed among multiple clades. And so they come up with the idea that this outbreaks isolate came about by reassortant. And their proposed model is that this virus, 2015 to 2024 clade, is a novel reassortant that acquired M1 from a virus circulating in 2009, 2018, and L2 and S from another virus clade. And they spend the rest of the paper building evidence for that and talking about how it spread. And I like these papers very much because they have words like a Bayesian Markov chain Monte Carlo framework. So you get to go to Monte Carlo to do this work? This is so cool. And so they want to know, when did this virus enter the human population? The time of the most recent common ancestor, which we call TMRCA. So the 2015-24 clade, they reconstruct the time-scaled phylogenetic tree. And so this OROV-BR for 2015-2024 seems the TMRCA seems to be 2013-2014, which is slightly before the sampling time of the oldest sequence from that clade. So that would make sense. And so the time frame of the reassortment events is between 2009 and 2013. And then they try and do a more finer reconstruction of the reassortant events. They have a model. These are all based on models of the sequences, right? This is the coalescent with reassortment constant population model, the COLRE model. And then extend with the Bayesian Markov chain Monte Carlo framework. These are all statistical applications where you say, what's the likelihood that this virus originated from this at this point and so forth. So they say from this they find the reassortment event that originated the 2015-2024 clade, which is the one we're talking about making all these outbreaks, around 2010, which agrees with the analysis they just did on the segments, right? All right, so that's around 2020. This virus arose, and then it's you don't see it for a while, right? And then all of a sudden you get these outbreaks later on. So it's probably spreading and amplifying. So next they want to do the geographic analysis, phylogeographic reconstruction. So what sequences were present where in Brazil? So again, this clade 2015-24 was spatially and temporally segregated into 6 clades or lineages, right? I'm going to try and go through some of these, but I might confuse you. So there's a subclade RR1. It's all sequences from Roraima from August 22 to May 23. Then we have a macro 1, which is sequences sampled in this a macro region that Alan just mentioned before, right? Amazonas, Acre, and Rondônia states between December 2022 and May 23. Then they have another subclade, a macro 2, all the sequences in a macro from December 23 onwards. And they have 3 others, AM1, 2, and 3, which these emerged in the city of Manaus between mid-August and early September 2023 and spread to other municipalities in the central and northern regions from 2023 onwards. So the clade 2015-2024 was initially spread from the Amazonas State Central region following 2 main routes. There's a northward route where the virus reaches Roraima and originated the RR1 subclade that I mentioned already. That drove multiple outbreaks, and then that stopped. And then there's the southward route. The virus reached first Rondônia in August 22, and that's the origin of the AMacro1 subclade that I talked about. And from Rondônia, it spread to Acre and the southern region, causing an epidemic in different municipalities during the rainy season. And this clade persisted throughout the dry season of 2023, gave rise to 4 subclades found in the 23-24 rainy season. And this AMacro2 probably emerged in Acre around August 23 and spread to Rondônia and the southern region of Amazonas. And then I said that these AIM1, 2, and 3 emerged in the city of Manaus from that. So you see, they can really pinpoint very specifically where, by looking at the genomes geographically, where they were isolated, the routes have spread because, they have characteristic sequences, right? Okay, they try and get a feel for the population dynamics and dispersion rate of these interesting of these individual lineages, and these models they use a Bayesian sky grid model. They find an expansion of the population size from November to March in the RRA macro regions, and a decrease between April and October, which corresponds with the rainy and dry seasons. So there's a big bottleneck between the rainy and dry season, right? You have lots of reproduction in the rainy season, big population size, and then it collapses. So there's a bottleneck. Only a few viruses are going to make it through that. So basically, they think the seasonal pattern of these cases was driven by rainy seasons, a lot of transmission in rainy seasons, and low, but not zero transmission during dry seasons. Then they try and say, well, what was the speed of dispersion? This is very cool. And so they find the average dispersion rate of this virus is 1 kilometer per day. Gosh,
which is roughly within the flying range of a midge, apparently.
It's actually, it varies a little bit in the different states. It's 0.66 kilometers per day elsewhere. And then they find it temporally from August 22 to March 23, So it starts out at 1 kilometer today, and then per day, and then it goes up and reaches 1.65 kilometers per day by the end of December 2023. Then they find that there are actually short, medium, and long dispersions. And they find that and these have different percentages. They're very short, less than 2 kilometers, most of the dispersal events. That's probably by midges. But many of them, 2 to 10 kilometers and higher, are probably linked to human dispersal.
Infected humans going from one place to another.
Yeah, getting on a train or walking or a plane or whatever, going
boat.
And so the proportion of long, over 30-kilometer distance viral migrations in the Roraima state and Amacro was slightly higher than the Amazonas.
Those are not midge flights.
No, those are not midge flies. I thought that was cool that they can distinguish between the vector speed and the human assisted. So that's the data. Very cool. So we have this novel reassortant that emerges, and it percolates for. We don't even notice it. It spreads for a while, and then all of a sudden it explodes, and it's fueled by seasons of rainfall. And they say that rainfall replenishes the soil with moist sites suitable for oviposition and immature development, and the emergence of adult midges.
Yeah, so this is the essence of this virus picked up genome segments from widely separated areas, apparently. This genome segment was circulating over in one area, and another genome segment was circulating in another area, and the virus got transported, probably by human cases or humans moving animals, from place to place, and then you got this reassortment. The reassortant, for whatever reason, has become quite successful and has spread across this whole large region, much larger than a normal Oropouche epidemic, and has managed to, not over winter, but over dry season, survive into the next wet season. And I guess it's too early to tell if it's going to show up in this wet season, but I wouldn't bet against it because you've got thousands of cases there. And they also talk a bit in the discussion about some of the factors, the big picture factors that might have caused this to happen now versus, the past 50 years of these epidemics that we've seen. And there have been some big changes in Brazil and worldwide. There's climate change going on. There was an unusually severe El Nino event that sent the weather wacky in Brazil and many other places. And so that's going on. And also from 2019 or so till pretty recently, there was a significant expansion in rainforest cutting in this Amazonas region. And that was under the Bolsonaro administration in Brazil, which drastically opened up logging to billionaire friends of the president is one way of putting it. And that probably contributed by moving more people into those areas to do the cutting, by cutting down the trees, by increasing human to wildlife contact.
Yep.
So all of that fed in, all of which is human activity, and gave us this.
They call it the weakening of environmental governance in Brazil.
Yes. I was looking at that part really closely because I was curious about the survival through the dry season, and whether or not that dry season was unique, or at least unique in a way that they could comment on. Perhaps it wasn't so dry, and that was why it was easier for the virus to have persisted through that season. And unfortunately, because the world does not work as simplistically as I might think about it, there isn't sort of this obvious change. And they talk a lot about all of the different factors and how all of these things were going on during that time.
Yeah.
This is where the word pluviosity
Yes.
And they say these findings suggest a potential indirect effect of pluviosity on the increased transmission of ROV in the Amazon region. So just to remind you, this virus, it got M1 from a virus called BR 2009-2018. It was detected in the eastern Amazon region between 2009 and 2018. And the L and S2 segments from a virus that was detected in Peru, Colombia, and Ecuador. Not in Brazil.
Viruses don't respect borders.
No.
And these viruses are always reassorting, and one is just going to randomly show up that is really good, really has good fitness, let's say. It reproduces well and can transmit well. And that's the one that's going to take off. And that's what happened here. And then they say remember that reassortant events happened in about 2010. And so they say there was a silent dispersion for many years, and then it was detected in Tefe in 2015, a small inner city, and a remote rainforest village in French Guiana in 2020, and probably disseminated concurrently from Amazonas, both northward and southwards, reaching the Amacro region around mid-2022. So it's very interesting that you can track this so carefully, but also it just shows you that viruses are constantly undergoing change and silently transmitting. You don't have any idea what's going on until you see disease. And they say we need to do better surveillance to see what's going on beforehand, right?
Good idea.
And this applies to any virus.
Mm-hmm.
But this takes foresight. Many people who control the money don't think we need to do this. Let's just wait for a bad thing to happen. It's just really, we could make inroads at preventing this if we knew what was going on.
One thing they mention is a reference in another paper that Oropouche was successfully established in southeastern and southern Brazilian regions and the Caribbean region by early 2024. So it is already way beyond the Amazon.
It's already busting out. Yeah.
Have there ever been cases in the U.S., like imported?
Don't know.
109 cases imported from Cuba.
Right.
Mostly in Florida. Did you work on the virus a bit, Kathy?
So we got the virus when it was still BSL-3, so we went through BSL-3 training. And then by the time we got the money, it was then BSL-2, but then we didn't have the people. And so someone did come from Eurico's lab and grew up the virus and did a project in Akira's lab. But and we did make a virus stock here, but sadly, no.
Also, interesting point here.
First you get the virus, then you get the money, then you get... No, that's a different...
Sylvatic mosquitoes in Brazil may move up to 3 kilometers a day, and Culicoides species, Culex in North America, have a mean dispersal rate of 2 kilometers per day and typically travel less than 5 kilometers from the breeding sites. Interesting. Okay, I found that very interesting. So hope you learned about Oropouche and now also what you can do with sequence and geographical and temporal information, right? 3 things: the sequence, where, and when. And then you can establish what's going on there. Very cool. Okay, let's do some email. Alan, can you take that first one?
Sure. Benjamin writes, Dear TWiV team, greetings from Oslo, Norway, and thanks for another interesting episode. Uterine endometrial cancer is not necessarily caused by HPV. Studies are inconsistent and geography may be important, but most studies suggest a limited or no role of HPV. Self-sampling seems to be done in 5 of... So this is referring to, I forget the episode number, we talked about HPV, which is implicated in cervical cancer, but Benjamin's saying not uterine cancer. Self-sampling seems to be done in 5 of the 21 regions in Sweden, though maybe a moving target, and it can still be done in office as I understand it. It might have improved the screening rate and be cost-effective. Importantly, to eliminate cervical cancer involves continuing with the cancer screening, because what they usually find is either a precancerous lesion or even just an asymptomatic HPV infection if the PCR is positive but there's no visible lesion yet. In terms of avoided office visits from the self-sampling, you mentioned that there is other useful stuff that would be done during a checkup. I am coming at this more from an internist's point of view, but routine visits for no acute reason every 2 years or so are considered unnecessary by many, especially for younger people. Most guidelines recommend ceasing to screen for cervical cancer around age 65. How useful a visual inspection of the vagina and vulva is remains unclear. The pap smear with or without HPV testing is the most important part from a public health standpoint. Also, many, if not most, pap smears are done by gynecologists, and they often do not follow all the stuff that is recommended by, for example, the U.S. Preventive Services Task Force, say screen appropriate patients for diabetes, lung or colorectal cancer, or aortic abdominal aneurysm, or offer statin medications to selected patients. Yours sincerely, Benjamin.
It's interesting. I think last week we had a letter or sometime, maybe it was for Daniel, I don't even remember, where they said, you can, what was it? You can pick up some other kinds of cancers, but it's not worth the doing it because it's so rare. It's kind of an incidental finding, right?
Right.
Okay. Well, this is all based on a TWiV I did in Sweden where the doctor said, we're just doing PCR now. If you're positive, you do a self-swab, or you can go to an office if you want. If you're positive, they look and see if there are any lesions. So as he says here, you still have to do an exam of some kind. I guess you do a pap smear then to see if there are any abnormal cells if you're PCR positive. Anyway, thank you. Brianne, can you take the next one?
Sure. Anne writes, hello, Dr. Racaniello. So appreciated the diversion re snow walls in Japan on today's TWiV. The videos and other info provided by your listeners was a treat. I've never been to the snow walls. I lived in Tokyo for 7 years, but skied elsewhere in Western Japan, and the snow was always impressive. At any rate, I heard your comments about hokusai, and I thought you might be interested in an exhibit that is coming to my neck of the woods. Should you happen to make it to the Chicago area for this, please be in touch, as I'd enjoy meeting up with you. Other TWiV listeners in the area would no doubt enjoy it too. So please let us know if you're coming this way. Regards, Anne. And Anne gives a link to a hokusai exhibit that looks like it is between May 31st and September 21st, 2025.
Yeah, if you remember the thumbnail from that TWiV, it's right there, that first image, the wave.
Mm-hmm.
It was all about viruses making waves. And in that episode, someone sent us these big snow channels that they make, and I guess subliminally I picked the Japanese picture for that. Kathy.
Okay, so this is Katie, Vincent, Daniel, and team, writing from the Denver metro area where it's a clear, cold 19 Fahrenheit. A note of thanks to you all for your excellence in science communications via podcasts. I'm the daughter of a now retired radiation oncologist and had the good fortune of growing up around interesting science-minded conversation at the dinner table, even though I didn't go into that sector myself. Thank you for your consistent pushback on, quote, wellness industry nonsense and mis- or disinformation. And thanks for taking the time to sit and record in the first place. You have saved my sanity many times as I look around at the worst timeline in the USA with disbelief and think to myself, am I overreacting? And I realize I'm not. I'm just informed, and my general social circles are not super science-minded. Your platforms, already vital, become even more so now. I'm happy to be a new Patreon subscriber and supporter. As an aside, I heard the most recent TWiV when you shared about the death of your friend and colleague Dixon, sending my sincere condolences to you all. Sincerely, Katie. So thanks for becoming a Patreon subscriber, and thanks for your remarks about Dixon, Katie. Nice.
Guys, do you think the plan is to make America sick, and then we can't do anything when they take over?
No, there isn't
That
like
would-
some genius plan.
Yeah, I was going to say that would imply that they have a plan.
Yeah.
Well, they have
Yeah.
this 900-page thing, right? The
This is
manifesto.
just about causing chaos and trying to drum up the culture war BS, and it's, yeah. That's really all there is.
I hope you're right.
Well, that's bad enough. I mean, that's
Yeah.
giving Elon Musk the keys to the treasury and all that stuff is these are going to have bad impacts. These are already having bad impacts.
Yes. Oh, let's just turn this off. Let's turn this off
Yeah.
without planning and telling people a few months ahead of time, right? That
Yeah.
would be the normal thing to do. Why can't they do that? Why can't they
Because it
say,
wouldn't cause as much chaos. It wouldn't be as entertaining. It wouldn't make good television.
Let's stop funding the USAID or PEPFAR and have children die.
Yeah.
I mean, they must know that that's going to happen, right?
No, they don't care.
Yeah, I was gonna say, do they care? And
No.
that's what's really sad.
Aldo writes, I extremely enjoy all the episodes of TWiV. I listen to them driving to and from work. In episode 1199, there was a discussion of biosafety levels and indicating that international organizations do not have standards. The WHO's biosafety manual can be found at, and Aldo gives
I
a
don't think
link.
that's quite what we said, but all right.
However, they moved from biosafety levels to a risk-based approach. But in previous version, they still had the biosafety levels. And gives a link, I think, to that. And then there's a table here, risk group 1, 2, 3, and 4, right? Classification of infective microorganisms by risk group. So risk group 2, a pathogen that can cause human or animal disease, but it's unlikely to be a serious hazard.
So
to
these four
workers.
groups are based on individual risk to the
Yeah.
researcher and community risk if it gets out. And so
Yeah.
you could, if you have low risk at both, then you're group 1. If you get moderate individual risk, low community risk, you group 2, and then moving on up.
Yeah. Yeah, this is because of our discussion of the coronavirus paper, which they did at
Yeah.
BSL2, if you remember, right? Anyway, risk group 2, you might get sick, but effective treatments are available and so forth. All right, so returning to Aldo, the virus that was handled in China had a moderate individual risk and a low community risk. Although the virus was present in bats for presumably a long time, there were no human infections. There are in humans a large number of coronas that can cause infection with a low or moderate individual risk and only 3 to 4 with a high individual risk, and perhaps only SARS-CoV-2 with a high community risk. As scientists, we should do the risk analysis and not assume a zero risk attitude. So, in my opinion, the classification of the Chinese laboratory was correct. It's easy to say that to prevent possible huge consequences, we classify it as BSL-3, but that's not science-based, and we should stick to the science. The VSV virus was presented as safe, but I did a pig infection study in a BSL-3 animal facility with air protection. But the pig did not sleep that well, and during intradermal injection in the snout, it moved, and the needle ended up in my finger. 2 latex gloves do not protect against needles. I had a fever the next day, and 2 weeks later, a bacterial infection in my tendons, which caused problems for 9 months. In hindsight, our biosafety officer should have put me on antibiotics because the needle had been in the skin of the pig. Right. Sorry you went through that. BSL-3 only has an additional protection against airborne infections, and many viruses, especially those from the gut, do not spread airborne. We should not overestimate the safety of a BSL-3 facility. In a BSL-2 facility, most of the airborne infection is controlled in a Class 2 safety cabinet. Aldo is project leader in vesicular diseases at the Wageningen Bioveterinary Research, which is in the Netherlands, I believe. Wageningen, right?
Mm-hmm.
Yep.
Thank you. I appreciate those thoughts, Aldo.
Mm-hmm.
Yeah,
yeah.
I am not entirely convinced that the virus in that paper was a low community risk. It was an unknown community risk. And I certainly still stand by my argument that if you are the Wuhan Institute for Virology, maybe the higher standard should apply just from a public relations standpoint.
Oh, that's because of the history there,
Yeah.
right? Yeah.
Not the actual history there, but the
I got it.
Yeah,
got it.
the
But
perception.
maybe somewhere else. it wouldn't
Maybe
be
somewhere else you could do this at BSL2 and get away with it, but I
Maybe
think
the
it was
phylogeography of the Wuhan
Yes.
Institute.
Yes. All right. We just have two more, so let's finish them. Who started?
I
Brianne?
started, so I'll go ahead and
Okay.
take David.
Go ahead.
Hello, I'm the author of Vincent's Pick of the Week in TWiV 1203. This is science-based medicine use of placebo controls in clinical trials, why the value of placebo controls is not an affirmation of a powerful placebo effect. It is gratifying that you found my article useful. Thanks for the shout-out. I have tried to volunteer some of my time to good science communication in a time when it is sorely needed. Science-based medicine has afforded me a great platform to do so. It's nice to know that other science communicators are noticing. Best wishes, and it's David Weinberg. Thank you, David.
Yes, I enjoy science-based medicine very much. And thank you for writing that. Everything I read there, I really enjoy. And Brianne.
Sure. Walt writes, I think approximately everyone, including science deniers, acknowledges that flu viruses mutate, and in so doing may become more pathogenic in humans. So I think you should call it out. Our dangerously wrong-headed HHS secretary is calling for biosafety level 0, gain-of-function quasi-experimentation that could accelerate the next pandemic's onset.
Let the virus rip through chickens.
Yep.
Yeah. Well, this is what happens when you don't know any science. And, you know,
And
the
don't
head of
care.
the Department of Agriculture is a lawyer also with no science background. So this is what happens.
Well, I'm not sure that saying they know no science is completely correct. I think they know enough science to be dangerous.
Yes. They don't understand science.
Well,
Or don't
If you
care
know
to.
science, how can you say that vitamin A is the cure, but when the literature says it's not, right?
Well, it depends what you mean by no, but
Yeah.
you know, it's not that they know absolutely no science.
understand.
It's that they're just
But-
not.
Listen,
Yeah.
if I ever get an opportunity to speak in public, I would say they know no science, just because there are people out there that don't get the nuances even,
Right.
like
Right.
you are, right?
Right.
Okay, time for some picks of the week. Brianne, what do you have for us?
So I have an article about something that I've been spending a bunch of my free time thinking about, which is NCAA basketball and March Madness and brackets. And so I have an article from Smithsonian Magazine about the math behind having a perfect bracket. And so picking an entire bracket perfectly is a 1 out of 2 to the 63rd power chance. And we think it has never been done. And in fact, it seems like it probably would never mathematically work to have been done in the history of the universe. It would require so many different options. And so this is an article kind of talking about the math of that. And I think that even sometimes I would argue, but there's some that those possibilities that aren't really realistic. You could probably figure it out, but they go through this and you say, well, if you assume that this is going to happen or this is going to happen, this is how that would change your probability of picking a perfect bracket. And in the end, this really tells us that picking a perfect bracket is very unlikely to happen, but it's sort of a fun way to think about about probability and how rare events these things are. And I enjoyed reading it.
So perfect means you get all the winners right.
Get all the winners
Yes,
right. in
all the winners
games.
right for all the rounds in all the games.
Yeah.
Okay.
Neat. My approach, the few times that I actually did any kind of a March Madness thing, I've never bet money on it, but I just went through and picked which mascot would win in a fight.
Yeah, no, I know people who pick that way and who pick with all sorts of fun ways. I will
Yeah.
admit, my best upset this year, I picked because I attended a conference at that university. It was very cold there, so I had to buy a sweater at the bookstore. So I randomly have a sweater from that university. So I decided, and I like that sweater. So I decided that was a reason to pick the school
pick
when I
that
had no
team.
other reasons. And they had some upsets. So I felt very excited about my sweater's power.
Kathy, what do you have for us?
Well, at least 5 regular TWiV listeners know that I have signed up to do frog and toad surveys for the city of Ann Arbor. Went to a 2-hour training a couple weeks ago, almost a month ago, where we were told how we would be able to listen to the calls and identify them, and over what stretch of time we should expect which frogs. And then we could preference different routes around the city and go out and listen for them. And so now I've gone out to the places on my route. I've heard one frog because it got warm and it was a rainy evening, which is like perfect. You have to go after sunset. And then it got cold again. So the volunteer coordinator is so enthusiastic. You know, he was apologetic that he got us all set up for hearing lots of frogs, and then it was too dry, and then now it's gotten cold again. But he sent us these 2 videos about wood frogs, which can completely freeze. They put urine in their bloodstream and sugar in their bloodstream, makes them basically make their own antifreeze, and they freeze completely solid, and then they thaw and they're alive. And so he sent 2. One is from PBS and one is from Smithsonian. So he said, take your pick, the calm PBS version or the dramatic Smithsonian version, or enjoy them both to reflect on different communication styles. And each one of them is less than 4 minutes long. So I highly recommend that you watch 1 or 2 of these.
These are fun.
Yeah, and see what these
Cool.
wood frogs can do. And so they're one of the ones that's out now. And while we've been doing the show, it has warmed up to 61 degrees Fahrenheit, which I'm excited about because that's 16 Celsius, and the sun came out. However, that means that it didn't rain today. And so tonight's supposedly good evening for frog listening may not pan out, but
Might be
I'm
too
going
dry.
out anyway. Yeah.
I hope they don't croak.
Funny.
Alan, what do you have for us?
Actually, I was looking through my picks, and then I saw Kathy's pick, and so that actually inspired me to cue this one up. It's another springtime topic. This is about polar bears, baby polar bears emerging from dens. And I apologize, you will not see a cute, fuzzy baby polar bear close-up coming out of a den. But this article in Science talks about how difficult it is to study polar bears in general, and in particular to track the birth of new cubs, because this happens in these brutally cold areas. And I mean, the mother bear itself is a deadly dangerous predator. And so these scientists who are studying them used a combination of GPS collars and remote video, which is up. They found, I guess, a high location on a mountaintop looking down at where this mother bear is denning. And then you see the time-lapse footage. She's going around digging a few different holes in the snow and then denning. And then as the season progresses, you see her come out and run around a little bit, and then she comes out again and runs around. And if you look closely, there are a couple of little fur balls right behind her. They're really, really close to her, and they're following right along.
It's cute. I really liked watching this.
Yeah.
Mm-hmm. It's a new word in the Oxford English Dictionary that means impossibly cute. Did you hear?
No.
Okay.
What is it?
I'm gonna look it up. Gigil. Overwhelmed by cuteness. Gigil. So it's not translatable. So
Giggle? Jiggle? Jiggle?
I don't think you pronounce it giggle.
And there's no etymology for it then either, I guess.
I think it's from a different language, but I don't know. Anyway, Gigil. Impossibly cute.
It's a Filipino word.
Yeah.
Okay. So it's a Tagalog.
Yeah.
Gigil or Gigil.
Just
Gigil.
doesn't fly off the tongue.
Gigil. Okay.
My
Now-
pick is
Now that we're talking about impossibly cute things,
yes.
I just checked and I realized no one has actually picked yet, and so now I'm really sad. The woolly mice. Someone should pick the woolly mice.
Oh my gosh. Yeah, I blogged about this.
They're so cute.
They are really, really cute. It's
Hmm.
it's just, I'm sorry, it's a ridiculous project, but
it is, but they're cute.
but they're really adorable.
My pick
You
is
heard about this, Vincent? The woolly mice?
I didn't know. I didn't hear.
There's this company that wants to resurrect the woolly mammoth. That's the part that's ridiculous.
Mm-hmm.
But as part of that, they did this multi-gene insertion mouse. That it's a mouse that produces a woolly fur, very much like a woolly mammoth is presumed to have produced. And the mice are very, very cute. They're
Cool.
these super shaggy woolly mice.
Okay, my pick is a Wired article by Tomonari Kotani. It's called Hikaru Utada Would Rather Play CERN Than Coachella. So this is an interview with a Japanese singer-songwriter who was fascinated with science. The names of songs on her latest albums include Schrodinger's Cat, Quantum Entanglement, right? So she's fascinated with science. So she says, she said, CERN is a place I have dreamed of visiting for the past 10 years or so. To be honest, being able to go there and talk to the scientists and see the particle accelerator might be even better than performing on the main stage at Coachella. So Wired brought her there.
Nice.
And she got, so they talk a little bit about CERN. And then she got to interview one of the scientists there. So I think this is very important that popular people should embrace science, right? Here we have
Yes.
a singer-songwriter, and she understands the value of science. So get with it, people who follow singer-songwriters, right? I think this is really good. So
Cool.
Nice.
I'd rather play CERN than Coachella. It's very cool. That's TWiV 1205. You can find the show notes at microbe.tv/TWiV. Questions, comments, picks, TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. Let's see, what is it? Microbe.tv/contribute. Say it every week, twice a week. Can't remember. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, Bio-Prof Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot. I really want to read more about these wood frogs now.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv if you want to go to Blue Sky. We do something over there. Microbe TV, no dot. That's the address on Blue Sky. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamp. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.